IDH1, a biomarker of hope for glioblastoma patients. Spoiler alert, cancer kills. Cancer kills up to 1 in 6. IDH1, a biomarker of hope for glioblastoma patients. Spoiler alert, cancer kills. Cancer is responsible for up to 1 in 6 deaths globally every single year. Traditional hallmarkers of cancer include resisting cell death, sustaining proliferation signaling, evading growth suppressors, activating invasion and metastasis, enabling replicative immortality, and inducing angiogenesis. These hallmarkers are a classic of just about every cancer and are literally what make cancer cells cancerous. So, for example, in enabling replicative immortality, oftentimes cancer cells have an enzyme that will create telomeres at the end of their genome to prevent genomic damage. They also do things to resist cell death and evade growth suppressors by literally hiding from T cells. Maybe they put certain ligands on the outside, or they do things like preventing caspase-3 from going through their typical apoptotic pathways. And another major hallmarker, which is inducing angiogenesis, sustaining proliferation signaling. Obviously, it takes a lot of energy to be able to grow unchecked. And so these are some ways in which cancer cells are able to do that by creating vascular pathways. And they basically send out signals to the body saying, hey, we need more blood flow here. Hey, we need more blood flow here. Um, and they recruit the cells around them to develop this microenvironment that is beneficial to the tumor cell. So specifically glioblastomas, which is a type of brain cancer, are generally fast growing and fairly aggressive. They're considered one of the most aggressive cancers when it comes to the central nervous system. They often invade nearby brain tissues, but they rarely spread to other organs. So oftentimes, even if there is a localized tumor, around that will be cells that invade into the tissues around it rather than staying localized into the tumor. Most prevalent of the brain cancers or central nervous system malignant cancers, and it accounts for around 48% of all cases. So, pretty large, almost half of the cases are due to glioblastomas. And as I mentioned, it's pretty aggressive. So if the cancer has gone undetected or untreated, it's typically fatal within 6 months of development. Overall, there's a slim survival rate post-diagnosis too, approximately 40% um, in the first year will survive, and then only about 17% in the second year. So their challenges are just as complex as the cancer itself, right? So the brain has a limited capacity to repair itself. So even when you are treated with chemotherapy or radiation, obviously those treatment plans can't necessarily target only cancerous cells. So any tissue that either the cancer has taken over or some of the tissue that the treatment has killed off has a hard time repairing itself. Oftentimes, there are tumor-induced seizures, and that results in further brain damage. Localization of tumors often leaves surgical removal impossible or too risky. Obviously, the brain is a very complex and sensitive organ. And so surgeons have to be really careful and mindful of where they cut. And we've seen evidence of that in our infamous lobotomies, right? The blood-brain barrier presents challenges for drug diffusion. One of the key characteristics of the brain and its preservation is the blood-brain barrier, which is basically the tightest borders you've seen. And so very little other than glucose gets in there. And so that presents a challenge for drugs. Oftentimes, drugs are large or polar. And so being able to get treatment into those spaces can be a challenge. Disruption in typical blood flow presents challenges for effective drug treatment. So, due to leaky junctions, there's often, edema, swelling, changes in the blood circulation due to angiogenesis. It can overall cause some issues in the treatments. So, diagnosis in the patients, they can experience a myriad of symptoms. It typically includes persistent headaches, vomiting, double vision, loss of appetite, change in mood or personality, and or thinking and reasoning skills, new onset of seizures, and then gradual onset of speech difficulty. Um, Typically, CT scans and MRIs are common diagnostics technologies. You are also able to do a tumor chemical profile using MRS, magnetic resonance spectroscopy. So current treatments, as most people know, they can either include some sort of either biopsy or tumor removal. Intraoperative MRIs are useful during this because it actually allows the surgeons to see exactly where they're cutting. Oftentimes, it will be done while the patient's awake, so that they can evaluate their cognitive and speech abilities as they are moving around in order to limit the damage that's done during the tumor removal. Um, Post-surgery, it's pretty standard for radiation to kill any of the tumor cells that have invaded healthy tissue. As I said before, glioblastoma is pretty common to have that invasive tumor cells around it, and so it makes it pretty impossible to entirely cut out the tumor. But as previously mentioned, radiation can damage both healthy and cancerous tissues. And so iterations of these treatments often include 10 to 30 treatments 5 days a week. So I'm sure you can imagine the harmfulness that can be created to healthy tissue. And then chemotherapeutic drugs, those are often administered during radiation and preceding it in a 28-day on, 23-day off cycle for around 6 times for maintenance. So specifically IDH mutations, isocitrate dehydrogenase enzyme mutations have been seen in nearly 80% of secondary glioblastomas versus 6% in primary glioblastomas. So a secondary glioblastoma is a glioblastoma tumor type that has progressed from a lower grade to a higher grade. Um, So, clinical features of this type of phenotypic clinical features from the mutation include oftentimes a younger age prognosis. So, oftentimes patients are diagnosed at a much younger age. Um, there's a frontal lobe localization tendency in tumors. There is less frequent tumor infiltration, so the tumors don't spread as often or as much. And typically, there are more favorable outcomes, including longer survival. The IDH1 R132H, which is the arginine to histine at location 132. This makes up around 90% of all IDH mutations. So as we can see in the graph on the left, upper left, the low-grade glioblastomas percentage of cases affected is 70%. The next one is just glioblastomas in general, and still they are the second highest. Even though there's a huge difference there, they are still the second highest of cancer types to have this type of mutation. And then in the lower left corner, we can see that these mutations have a iteration as far as survival rates. So patients who actually have this missense mutation often have greater survival rates than those that do not. So the hypothesis here is that the R132H can be used as a biomarker to determine treatment plans. So the presence of the IDH mutation is a strong biomarker associated with positive prognosis outcomes, regardless of age or tumor grade. So the most common mutation, again, which is that IDH R132H, can be detected with anti-IDH antibody and immunohistochemistry. But in the case of negative or inconclusive results on suspected eligible tumors, DNA sequencing by PCR or next generation sequencing are effective. So up here we can see the difference between a wild type and a mutant IDH glioblastoma. So on the left, we have a negative immunohistochemistry, whereas on the right, we see a lot of this brown. So that is indicative of the mutant. So healthy metabolic pathways um, in the Krebs cycle, also known as TCA or citric acid cycle, this is where a lot of the metabolic pathways, in order to release stored energy from nutrients, kind of really kickstarts after breakdown of glucose. There are many key players, so there's a lot of different enzymes that are in there, and a lot of essential products that either go in or come out of, including amino acids. Um, uh, Involved in this process. So the isocitrate dehydrogenase is an enzyme that typically converts isocitrate into alpha ketoglutarate, and that's done by reducing either NAD plus or NADP plus, and releasing CO2. So as we see in this diagram on the right side, isocitrate dehydrogenase is there in the middle, and we're moving from isocitrate into alpha ketoglutarate. So, not so healthy metabolic pathways is that when the mutated IDH, um, there's a few different types. So, it is a dimer enzyme. And so, if there is the wild type will typically convert isocitrate to alpha ketoglutarate. When you have the wild type, a heterogeneous mutated enzyme, what ends up happening is it switches the enzymatic activity and it reverses and converts alpha ketoglutarate into D2 hydroxyglutarate. And this is a huge signaling pathway that has been shown to lead to different fates of angiogenesis and tumor growth. So, and then lactate dehydrogenase A, LDHA, also is known to catalyze pyruvate to L-lactate, which allows for rapid cellular proliferation. So LDHA is often upregulated in cancer cells, yet in the IDH mutant glioma, it's actually silenced. And this is the most likely explanation as to the slow growth of gliomas with this mutant IDH versus the wild type. So if we go to reactome.org, we can start to look into these molecular pathway interactions. The URL is down here in the lower left corner, and you come to this main page at Reactome. And you can choose on the left side menu. There's a bunch of different options that you can choose here in the middle. You see this kind of phylogenetic tree. And so if we click on disease, and then we click on disease of metabolism, because of course this is an enzyme that is in cell metabolic function. And we click on abnormal conversion of 2-oxoglutarate to 2-hydroxyglutarate. We populate this IDHR 132 mutant dimer. And so we can see here that in this metabolic pathway, we have now gone into these different types of abnormal conversions. The summation, there's some explanations of different things like summation disease. But overall, it says the high frequency with which the mutation is found in surveys of primary tumors is consistent with the possibility that it's advantageous to tumor cells. So this is one of those evolutionary selective mutations that has helped this cancer to survive. And then again, seen in these glioblastoma multiformes. So in a lot of these astrocytomas. So if we click on expression, we can see that there is levels of expression across the body. We have this kind of heat graph here with expression levels of adipose tissue, adrenal, bone, all the way through zone of skin. And at the cerebral cortex, which is going to be your central nervous system, your brains, you can see that your the heat map is rather dark blue. So we're seeing a lot of expression in that. Now, if you click on this cerebral cortex, you can either click on the words or you can click on the blue box underneath it. It brings you to Ensemble, again, URL in the upper right corner here, and it brings you to the IDH1 gene, and it is located on chromosome 2. In this first track here, chromosome 2, we see it's all the way over on Q34. You see a little red box over there on the right, and then the region in detail. And we can see here again this red box outlining the area in which this mutation is occurring. So if we want to see some protein to protein interaction, so we want to see what are the types of proteins that are often associated with these interactions with the IDH1 mutant or wild type, we can go to string. Again, URL located on the upper left corner here. And we come to this home page. I have already typed in my protein name, IDH1, my organism. It's auto-populates to Homo sapien, but of course you can scroll through and choose something else if you want to. There's a lot of different options that you can do as far as like comparing within organisms to, variations of species. But if we click on this, if we search, click the search button, we auto-populate into this main menu here where we have a few different options. The first one that comes up is your network. So the IDH1 is the red circle in the upper right corner of the network. Protein network, and we can see a lot of the associated pathways within this network, a lot of the associated proteins that interact in the pathways. So if we click on the pathway data set, we can see our different relevant data sets in Homo sapiens that are associated with this. And so there's different, carbon metabolism, metabolic pathways, TCA cycles, as we go through, metabolic intermembrane space, and we can see here these different ideations of iterations of proteins in which that are commonly associated with the IDH mutation. Now, on this database knowledge, if we scroll down, we then see these predicted functional partners, and we can see the score based on a scale of 0 to 1 of association. And so our input here is this isocitrate dehydrogenase at the top, and then we have our predicted functional partners. So we're able to identify what are some of the most common um, protein partners, so to speak, in a pathway that may be either regulated by or affected by our mutation. And of course, we're going to see these, as you might notice, IDH3 in three different iterations. Isocitrate dehydrogenase does come in three different forms, 1, 2, and 3. Most common mutations is of course 1 followed by 2, but specifically we're covering that, so it's not a surprise that we would see that. Next, we will go to Genomic Data Commons, which is a cancer data set from the National Cancer Institute at the NIH. This is their homepage here. And once we get on here, we can click on Cohort Builder. And we can create our cohort here of these specific cancer sets. So down here, we would click on GBM, which is the glioblastomas, and then LGG, which is under projects, which is the low-grade glios, which are, as we will see, some of the most common glioblastomas that have this mutation. And so you can click on gliomas. In this case, it doesn't necessarily matter. There's only 11 that are not reported, and this is a glioma type cancer. But if you wanted to narrow that down, there's other ways in which you can narrow these down, but they were not important to running this initial data set at this time. And so once we create that cohort and save it, we will click on Analysis Center. And then we see this cool little core tool and analysis tool page. And there's a lot of different things here that we can do. I'm just going to go over 4 of them. But as you can see, there's BAM slicing, clinical data analysis, sequence reads, set operations, oncomatrices, gene expression clustering. So there's a lot of different options here. You can build a variety of cohorts, and so there's a lot of cool tools that you can take advantage of with this. The first one is that I'll cover is this gene expression clustering. And so again, it's a heat map. And you can see down in the lower left corner that red is associated with more associations, right? Versus blue is not as many. And so we can see these chunks of along the top corner, I'm sorry, along the top axis is patient cases. And along the left axis is genes, gene expressions. And so we can see these clusters of gene families that are associated with certain clusters of cases. And so this one is done to 1,000 genes. So it takes 1,000 genes, but you can edit these to get down to just whatever it is you're looking for. Next, we have our mutation frequency. So with this, we can see that there is on the right side an overall survival plot, and this was earlier in the PowerPoint, but an overall survival plot that the IDH1 mutated cases have a better survival rate over a longer period of time than the mutated or wild type cases. And the distribution of the most frequent mutated genes in the middle bar graph, you can see is that IDH1 has the most affected cases within the glioblastoma and low-grade glios. So this protein paint we can see what the most common um, mutations are. And so, as was previously stated, the R132H is the most common. 90% of the mutations are the R132H. But there are other more rare mutations in the same on the IDH1 in this same region. So we can see the distribution of that. And you are able to click on these and kind of delve into detail a little bit more. And next, we have cohort comparison. So I did two different cohort comparisons. The first one here is a wild type versus mutant comparative survival rate. So as we can see, so the wild type is in blue. The legend got cut off here on the because you had to scroll down, but so the wild type is in blue and the mutant IDH1 is in brown or orange, whatever you want to call it. So as we can see here on the left farmost bars is dead, then next is alive, and then not reported, missing, and unknown. So just looking at dead versus alive, we can see that these wild type um IDH1s have a much higher percentage of death than that of the mutants. And then this is a cohort comparison of the low-grade gliomas, wild type versus mutant. So these are specifically about those low-grade gliomas, which again is where we're going to see most of those IDH1 mutations. And so, as previously stated, we can see that the higher amount of wild type is blue, mutant is brown, orange. But as we can see, as previously stated, and as studies have confirmed, that a majority of the cases are diagnosed at a much earlier age. So typically glioblastomas, astrocytomas, brain cancers, 55 plus, right? But overall, these wild type mutant low-grade glioblastomas are seen at a much younger age here, confirming again that hypothesis. So, some distinctive genetic and clinical characteristics between the two, right? So, our wild type, we see this, how things go as far as what kind of metabolites are used for synthesis, how do they take care of radical oxidative states, what kind of histone and DNA methylations are happening, stabilization, cell differentiation, and then for the patients on a patient level, what's a median overall survival rate? How is radiation and chemotherapy affecting some of these? So this is just confirmation from this study that the wild type is a lot harder to treat. It's often a little bit more aggressive. Not survival rates are much lower. And in this mutant IDH1, in all of these, HC and K, we're seeing this different pathway that, yes, may be creating more of these ROS oxygenated species, reactive oxygenated species, more methylation, but we're seeing that this cell differentiation is different. We're seeing that this overall survival rate is much different, and that when we are able to target those, we have better chances of radiation and chemotherapy being an effective treatment plan. So, as I started to say, the potential to target this either this IDH1 gene. So there are several compounds that are directly inhibiting the IDH1 mutant enzyme that have developed promising results by reducing the D2HG. They reverse histones and reverse DNA methylations. And the exploitation of the R132H mutation has been seen with epitope vaccine treatments, and it has shown to increase survival rate in mice trials through T cell response pathways. Um, Wild type IDH1 silencing has been shown to significantly reduce glioblastoma stem-like cells in reoccurrences. So after treatment, if there's reoccurrent cancer cells, there's a significant reduction in the GBM stem-like cells when silencing this IDH1 wild type. And then also several studies have targeted the wild type IDH1, and it has enhanced the responsiveness to traditional treatments. So when they have targeted this wild type gene, they have shown that the responsiveness has increased in comparison to when it's free reign. So some limitations on this study and on these um, abilities are that there really is no non-invasive way to determine an IDH genotype. So typically it's going to be through a surgery. Um, IDH mutated gliomas are more likely to develop a hypermutation phenotype, which then worsens prognosis and limits treatment abilities. So even though initially an IDH mutated glioma would be a good thing, it does have a higher risk of leading to a really bad thing. Clinical trials can be hard to interpret due to ethical considerations in experimental design and cancer complexity. There is no one size fits all treatment plan. So, what might work in a handful of people is sometimes hard to generalize. So, generalizability becomes a limitation. And then, unfortunately, progress is inevitable. So, progress of the glioma, of the cancer, is inevitable in glioblastomas. And so, we're really kind of looking at quality of life, quantity of life. How do we reduce some of those symptoms? How do we increase lifespan? And it's definitely not something that it's a cure situation.